Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the course on computer aided drug design. We will start a new topic that is called ADME. A stands for absorption, D stands for distribution, M stands for metabolism, E stands for excretion. That means absorption into the plasma, distribution inside the plasma and tissues. M is the metabolism that is taking place in the liver and because of the presence of different enzymes. E is the excretion, that is throwing out from the body either through the various urine or through the inhalation and exhalation. Okay. So, I did show this in the previous class. So, we have the compound structure which we can play around with: molecular weight, hydrogen bond donor, acceptor, the lipophilicity or the hydrophilicity, the polar surface area, the pKa shape, and which affects the reactivity. Now, three things are connected with each other: the physicochemical properties, that means solubility, permeability, chemical stability, okay, and the pharmacokinetics and toxicity, that means the clearance from the system, the half life, the bioavailability, the lethal dose, that is with respect to the living system. And here with respect to the proteins, the metabolism, the transporter affinity, binding to protein, target affinity. So, all these are interrelated. Okay. So, we can change a few parameters or structural features or descriptors, but in the physical level they get modified, in the biochemical level they get modified, and in the living system also they get modified. Okay. There are a lot of free softwares. One is called Mol Inspiration, then the CRDD web portal, that is computer resource related to drug discovery, Osiris Property Explorer, Swiss ADME. All these softwares can try to predict many of these properties like solubility, metabolism, and the various ADME properties, stability, and so on. For example, if you take this first one, the Mol Inspiration, that is quite a interesting software. So, this is called the Mol Inspiration software. Okay. So, we can go into free online services. We can draw structures here. We can draw any structures, or if I have the smile notation I can download. For example, if I have metformin I can download, or I can draw a structure. Here I will show you metformin. If I search, okay, so this is metformin. Yeah, we can copy the smiles notation, okay, and then take it to the Molins, and then we can enter the smiles notation. Okay, so metformin is taken here, and it gives you a lot of properties. Log P, this is based on some software, total polar surface area, number of atoms, molecular weight, okay, number of nitrogens, OH, NH, does it violate certain rules, rotatable bonds. So, it has got 3 rotatable bonds as you can see here, 1, 2, 3, volume of this molecule. Okay. So, this is the molecule, it is copied from the smiles. I can predict the bioactivity of this molecule. Okay. So, does it bind to a G protein receptor, ion channel modulator, does it kinase inhibition activity, nuclear receptor ligand, protease inhibition, enzyme inhibition. So, it can do all those things, and okay, then we can even draw 3-dimensional structure of this molecule. As you can see, we can view it, and we can get the polar surface area. Polar surface area relates to nitrogen, right? So all the nitrogens will appear polar, as you can see here. Okay, this is the electrostatic potential on this. Okay, then we can put it in a dotted form, so it gives you a dotted form, or it could be in a tube form. So you can look at the molecule in the tube form. It can be wire form. This is the standard. Okay, so we can do all those things, or so from the zinc we can copy, we can get properties, or we can draw molecule of our interest. As a medicinal chemist, I feel that I would like to synthesize a molecule like this. Okay, and I want to know the properties of this molecule. Okay, so I put in some chlorine here. Oh, sorry. So I want to put another here, then I put a chlorine, just blindly I am putting something. So we can calculate properties of this molecule. So it gives you log P 2.89. So it's neither hydrophobic, too much hydrophobic, or too much hydrophilic, which is good. Total polar surface area is low because it's predominantly a lot of CH2s, carbons. That is why polarity is very low. Okay, solubility may be a problem. Number of atoms, molecular weight, it's giving all these details. Okay, we can look at the bioactivity of this molecule. So it gives you all this bioactivity. We can draw the 3-dimensional structure of this and look at how it looks like. Okay, the molecule, and then we can look at polar surface area. As you can see, only very little polar surface area. Okay, because of the presence of oxygen. So predominantly it is a highly hydrophobic system. Okay, so this is a very interesting mole inspiration, as it is called. We can do that. Another software, like I mentioned, CRDD web portal, we can look at that also. That is called a computational research for drug discovery. There are a lot of softwares which can do the predictions. Okay, as you can see here, we will not go too much into this. The third one, which is the Osiris property explorer, and again this is Osiris property. We will not go into that also. Okay, if you want to look at that front page, this is called the Osiris property explorer. Yeah, this is Osiris property explorer. As you can see, again we can look at many properties tools, molecular property tools. As you can see here, we can get the property tool. We can try to get molecular weight, fragment rates, drug design, predictions, solubilities, all those things can be done. Of course, Swiss ADME is also there. We can use the Swiss ADME and then try to get property. For example, metformin, we went to metformin, right. So we can get the metformin, go to Swiss ADME, enter the SMILES notation here, and then we can run. So Swiss ADME gives lot of information, as you can see. It is still working, and it gives you molecular weight, number of atoms, it gives you log P in different techniques, different types of softwares, measures log P in different ways. So it gives you an average or consensus log P in the bottom. It gives you total polar surface area, molecular refractivity, hydrogen bond donors, hydrogen bond acceptors, molecular weight, GI absorption, then blood brain barrier, then PGB, that is P glycoprotein. Does it act as a substrate for that? No. It gives you solubility data here. Some of these rules we will talk about later, and this also pains we will talk about later. Okay, so it gives you a lot of properties. Cytochrome P, does it go and inhibit that, and so on actually. Okay, and then bioavailability also must be there somewhere. Bioavailability 0.55, that means that is good, very good. Of course, metformin is a drug. So okay, so and this is also an interesting graph which puts in the lipophilicity, size, flexibility, and number of unsaturated and then insaturated, that because of and then it draws the molecule, and or if it falls within this pink, that means it satisfies all condition. If one of the properties falls outside, so you may have a structure which looks different. So ADME is also, the Swiss ADME is also a good software for predicting lot of properties, mole inspiration. So all these softwares are good for calculating properties. As we go along, we will see more of that. Okay. Okay. So what is this absorption, distribution, metabolism, excretion? So we are going to spend a lot of time on them. Okay. This is important for all drugs. Frequent causes of failure of treatment, frequent, because you are not getting effective level. If I am having a bacterial infection, my concentration of the drug at the infected site should be higher than the minimum inhibitory concentration of that bacteria, so that the bacteria gets killed. Okay, so that is called the effective level. Or it may produce toxicity. Sometimes the drug may be too much, concentration may be too much, it may be producing toxicity. Drug-drug interaction, if you have 2 drugs, 3 drugs given to the same patient. For example, for blood pressure they may give you a beta blocker and they may give you a ACE inhibitor, and they may also give aspirin for blood thinning. So you have 3 drugs, there could be interactions between these drugs. If we want to understand different dosage forms, sometimes the drug is given orally, sometimes intravenous, intraperitoneal, intramuscular, intravaginal, it could be through the skin, it could be nasal. So we need to understand how the drug gets into the plasma through different routes, what happens to the maximum concentration and so on actually. Okay, so we need to understand ADME, which is very important, and most, as you can see, 39% of the drug failure during clinical trials because of poor ADME. We realize that when we do animal trials and when we do human voluntary trials. Lack of efficacy, the concentration at the target site is not sufficient. If there is a tumor, the concentration of the cancer drug should be high enough to kill the tumorous cells. Okay. Toxicity, 21% of the drug fails because of toxicity. So you see, we need to address that, which is based on, as I have been telling, the bioavailability, how it gets absorbed in the GI, the solubility, the lipophilicity, distribution inside the bloodstream, whether it gets metabolized because of the presence of different types of enzymes, does it get metabolized because of different pH conditions, does it get excreted, even that is very important. Okay, so we will look at each one of them. This is how the drug travels. So imagine I am given a drug orally, okay, a tablet form. Okay, so the drug is given as a tablet form. So it goes into the GI tract, it goes through the stomach, pH is 1 to 2, then it goes to the small intestine, finally to large intestine, that is colon. Okay, so the pH keeps increasing, coming to basic. So drug solubility becomes a problem, drug dissociation happens and drug gets absorbed. Okay, so if it does not get absorbed, drugs get excreted. Okay, so some portion is lost. So it gets absorbed, it comes into the bloodstream, and then liver is a fantastic gatekeeper, so it starts degrading the drug. So if it gets degraded and some drug gets absorbed in the tissues, okay, some drug gets degraded, metabolism, excretion, kidney, lungs, so you lose. Some drug gets attached to the tissues, absorbed, so you lose. So only small amount of drug goes into the bloodstream and reaches the target site, and again only part of the drug will go to the active site, part of the drug will go and bind to other proteins, so that also becomes useless. So only a small amount reaches the target site. So I may be given a large concentration of the drug, but what reaches at the target site may be very less, and this ratio is called the bioavailability. So maybe 20%, 30%, 40%. Higher the percentage better, because I can give a small quantity of the drug, so toxicity is low. I do not have to frequently give the patient the drug. So bioavailability, larger the value, better it is. And as we saw in Swiss ADME software, you approximately get a feel of the bioavailability. Metformin, it gave a number of 55%. Okay. So if you go to the database like drugs.com, okay, it may give the actual value, because metformin is a real drug. Okay. So this is called the bioavailability. This determines the drug dosage, this determines even toxicity, because if the bioavailability is poor, the patient has to be given large quantities of the drug, so that it could be toxic. If the bioavailability is very good, patient has to be given smaller quantity. If too much drugs gets excreted, that means if the half life is very short, so the patient has to be given frequently the drug, that means the dosing times increases. So everything is determined by this particular picture. Okay. This picture gives you nicely what is happening in at different places as the drug travels, right from the oral cavity in the mouth right up to the target site. Okay. Now another thing that happens is the drug, when it comes to the oral mucous cavity, pH is 6.85, but as soon as it reaches the stomach, the pH, as I said, is very low, 1, 2, 3. So that means there could be a drug degradation because of acidic pH. For example, ester bonds can break because of acidic pH, amide bonds can break because of acidic pH. So we need to watch out. Then when you come to duodenum, ileum and so on, the pH goes up and up, so it goes to 5.8, okay, 5 to 8 sorry. When the drug goes to the plasma region, the pH is 7.4. Okay. So the drug stability, solubility, everything should be constant irrespective of the changes in the pH. Then it goes to the liver, where there is a lot of degradation, then it goes to heart, then a circulation system, lungs, arterial system and so on. Okay. Then it goes to the target cell membrane, then it has to get into the target site and becomes efficacious. So there is a distribution of the drug. It is like a big pot of water where you put small amount of salt, the salt gets totally distributed, concentration becomes low. If the amount of water in the pot is very large, concentration becomes very small. If the amount of water in the pot is very small, concentration is high. So it is not the volume depends not only on whether it is male or female, but also on the type of drug, because some drugs will remain only in the bloodstream, some drugs will also get absorbed in the tissues, so the concentration in the bloodstream becomes low. So it depends on the type of drugs. Okay. A very small molecule can easily get absorbed in the tissues, so the amount present in the bloodstream may be low, whereas a large molecule and hydrophilic might not get absorbed, so the concentration in the bloodstream may be very high. So for the same person, depending upon the drug, the volume of distribution, as we call it, can change. Okay. So it is not constant. So it can depend on the race, it can depend on the gender, it can depend on the type of drug, it can even depend on the food we eat and the health of the patient and so on actually. Okay. So as I mentioned, the drug comes in and travels, it gets absorbed into the liver, whatever is not absorbed goes as excretion, from kidney also there is excretion, from the liver it goes to plasma, it gets absorbed in tissues and finally reaches target site. So this concentration that was this, okay, the ratio multiplied by 100, this is called oral bioavailability and it is mentioned as F, okay, that is the oral bioavailability. This is a very important parameter to understand. Okay. So when we talk about absorption, there are many routes for administration of the drug, right. So it can be oral, sublingual, rectal, so it can be soluble form, okay. It could be enteric coated, so the coating goes away because it keeps the drug stable without degradation, or it could be a slow release formulation. So there are now slow release partially crystallized metformins are there, so it could be a slow release formulation. So the drug gets released over a period of time, okay. Parenteral, it could be intravenous, it could be intramuscular, it could be subcutaneous, intradermal. So different rates of absorption you are going to have, different plasma peaks. So if I introduce the drug orally, after about 1 hour 2 hours, slowly the drug reaches a max and then it starts falling down. Whereas if I give it intravenously, immediately you find a big increase in the concentration in the bloodstream, okay. Depending upon the route, the time at which the max happens changes, and the amount or concentration also will change, okay. Skin, sometimes skin patches, nicotine patches you have heard of, right. So all these matters. Lungs, through inhalation, like some bronchial drugs, local or systemic effect, like even some anesthesia, okay, through lungs. Vaginal, that again it becomes local, okay, treatment of a certain bacterial infection or fungal infection. So what is this subcutaneous means? That is in the fat layer underneath the skin, okay, and intramuscular means into the muscle, intradermal means into the dermis of the skin layer underneath the epidermis, okay. So epidermis is upper skin layer, so subcutaneous is into the fat layer, whereas intradermal just below, intramuscular into the muscle region. So you can have different types of parenteral routes. Of course eye, as you have heard of eye drops, they are again local, okay. So some of them are local, some of them could be systemic, okay. Most drugs enter the body by mouth or injection. They must cross barriers to entry, that is skin, gut wall, alveolar membrane, then they get distributed by the blood to the site of action, intra or extracellular, they have to cross the barriers. So it may have to go through capillaries, cell wall, they get biotransformed to several different compounds, because there are a lot of enzymes in the liver. Liver is a very fantastic gatekeeper, it starts degrading whatever compound is present. So you have esterases, you have hydrolases, you have oxidoreductases, you have lipases. So all these start degrading your material, your compound, and they may become either sometimes toxic, or they may become totally useless, inactive form, or excreted. So it can be excreted through kidney, different routes which removes them and or their metabolites from the body. So you not only the drug, even the metabolites may be getting removed. So pharmacokinetics is quantification of the whole thing. So what the body does to the drug is called pharmacokinetics, okay, and what the drug does to the body is called pharmacodynamics, okay. So one is called pharmacokinetics and one is called pharmacodynamics, okay. Let us look at each one of them little bit. Absorption, some drugs work outside the body like your creams, laxatives, okay, but even some antibacterial fungal at the surface, some enter the body, enteral, oral, sublingual and so on. Parenteral, okay, subcutaneous, intramuscular, intra and cross, they have to cross the lipid barriers and cell walls, gut walls, capillary walls, blood brain barrier, okay. So a lot of diffusion limitations happen, and then they get into the body after distribution to reach their target, okay. So what are the factors that affect oral absorption? Drug has to disintegrate, that means the tablet, if it is in a tablet form, it should break down, it should completely dissolve, that is dissolution. Drug has to be stable in that pH in the GI system, stability of drug to enzymes, there are so many enzymes which I mentioned about, motility and mixing in GI tract, they are mobile, they get mixed with the GI fluid, they may be getting mixed with food and so on. Okay. Presence and types of food, what type of food we have eaten, sometimes proteinous material can just absorb or adsorb drug active ingredients. Okay. The drug may get diluted because of presence of fluids inside the GI. Passage across GI tract wall, that means through the passive diffusion. So that means the permeability has to be reasonably good, which we talked about in the previous classes. Okay. Then blood flow to GI tract. So it depends on there is good blood flow in the GI tract. Gastric emptying time, because again I showed you in the previous class, the emptying time varies. For example, in the stomach it could be about only 3 hours max, whereas in the small intestine it may go to several hours, and then large intestine it could go into several days. So the emptying time matters. Formulation, how we have formulated the drug, the manufacturer has formulated, he might have added surfactants, stabilizers, solubilizers, so how it has been formulated. So all these matters. So typically you can look at if the drug is given as an IV, the concentration of this drug will increase with time and then fall down. This y axis is concentration, x axis could be time. If it is given orally, you will not reach that peak concentration, and also the peak will happen much later, because the drug has to solubilize in the stomach, get past the permeable barrier, lipid barrier and reach. So it will, the max time will change, and the level height also will change. Okay. So the bioavailability is area under this curve AUC oral, area under this curve IV. Okay. So obviously AUC oral will always be less when compared to AUC IV. So it will always be less than 1. So we can multiply by 100, and that gives you the F value or the bioavailability value. So that is how you can find out. If I have a rat and a drug, I give it through IV, I get this particular graph, then I mix it with this food and allow the rat to consume it orally, and I will get and monitor the concentration of the drug as a function of time by taking blood samples, and then I will get this graph. I take this ratio to understand the bioavailability of the drug. Okay. So bioavailability, absorption determine the proportion of the drug in a dosage form available to the body. Generally IV injections, we assume it gives 100% bioavailability. So it is the ratio of the area under the curve, okay, concentration through other route. But it is not an indication of the drug effectiveness, it is only availability. Please remember, it is not about the effective, whether the drug is effective, that is whether it kills bacteria or whether it kills cancerous cells, that you might have collected data in your in vitro studies. Okay. So the bioequivalence can change, okay, bioavailability or bioequivalence. For example, this is a very interesting data. This is a compound called digoxin, okay, and different manufacturers make this. Imagine 8 different manufacturers make it, and you give 0.25 milligram of that particular drug, but the plasma digoxin concentration could be very different. Okay, so much different, as you can see here, the mg per ml. This manufacturer you may have 3, whereas this manufacturer it could be 0.4, so almost 10 times difference. Okay, so the availability of this particular drug depending upon the manufacturer has changed so much. Why is it? Because of the way they manufacture, the way they make the tablet, the way this other components have been added, so it can change dramatically. Okay. So sustained release preparations, okay, like with oil, drugs are mixed with oil or made into viscous material or made into particles, so that it slowly degrades particles and releases the drug, or viscous material. So the drug slowly comes out of this viscous oily material, or multi-layer tablets. We can have a coating of different biodegradable polymers, okay, like a gelatin, chitin and so on. So, they degrade slowly, drug gets released slowly, multi-layer tablets. It can help in slow release, it can reduce the toxicity, it can also help in the drug stability, okay. Yeah. Sustained release capsules, so we can have it in resins, it slowly releases over a very long time. Infusers, okay, so we can have infused, the drug gets infused, sometimes we can have a sensor, it can check the glucose level and release the anti-diabetic drug, okay, that is called infusers, okay. So, it is kept inside the body and the drug gets released from time to time or based on certain conditions. Skin patches, you must have heard about nicotine patch. So, the drug which is in the patch slowly gets into the body, okay. Prodrugs, I explained, it is a drug and another molecule connected through a bond, the other molecule helps the drug to maybe cross the membrane, maybe improve the stability, but inside the body the presence of enzymes may degrade and the drug gets released and then it is in active form, okay. So, the prodrug is not active, but once the drug gets released inside the body because of some enzyme action, then the drug becomes active, okay. Liposomes, drugs could be encapsulated in liposomes, okay, they are highly hydrophilic systems. Targeted drug delivery, we can target to a particular site using certain cues based on pH or temperature or presence of some other enzyme, antibody directed. So, as you know, antibody antigen type of combinations, so the drug will go and bind to only one particular okay, antigen, not to any other antigen. So, a lot of different approaches by which drug could be targeted, okay, and also the release profile could be extended over a long period of time, okay. So, we will continue again in the next class on this topic of ADME. Thank you very much for your time.